So from DNA to proteins, let's look back down at those codons again, right? If I have a double helix of DNA, one strand might say GTA, AAA, GGC. And the portion of the nucleic acid strand is called a codon. These are 3 base pair sequences, and they tell the cell to make a specific amino acid. So the sequence GTA tells the cell to make histidine, and the sequence AAA tells the cell to make phenylalanine, and the sequence GGC tells the cell to make proline and to hook these guys to each other. So make a histidine, hook it to a phenylalanine, hook it to a protein. That's what codons do. Why are they in 3s? Well, there are 20 known amino acids. And so if I have 3 base pair sets and there are 4 nucleobases, that allows me to have 4 to the third, 64 unique codons. If they came in 2s, I'd only have 16 codons. If they came in 5s, that's way too redundant. And so we have 4 nucleobases, CATG, and we must have 3 base pair sets. They tell us to make 64 unique codons. There are 64 different ways to have 3 sequences. I told you there were 20 amino acids, and so apparently there are more codons than there are amino acids. And the way that works out is, take a look here, for example. You've got 6 different base pair codons that say make leucine. So if you see a TTA codon or a CTA codon, they're both telling you to make leucine. Look, there's 4 here that code for proline and 4 that code for alanine. Notice also that there are codons TAA, TAG to say stop protein synthesis. There's another one that says stop protein synthesis. There's a codon for start as well. So codons say start the synthesis, make amino acids. There's a codon that says stop as well. But they're redundant. That's okay, right? Because that says that maybe if there's a mutation in the DNA sequences, that I might end up with a similar thing. So if I had a CTT, but somehow I had a mess up in my DNA sequence and it was CTA, that's okay. I'm still actually making the same amino acid in the protein. So your body actually has some redundancy built in so that if problems arise, something like that can happen. That doesn't always mean it's the case, right? Say I had a CTT that turned into a CCT, a leucine into a proline. That might be a problem later down the road. But at least in this case, it's not. Or not in all cases.